Good morning, everyone, and welcome to the last episode of our PGC Talks about COVID series. I am Chilette Monson at Ishel, the Philippine Genome Center Stakeholder Engagement Specialist. PGC Talks 2021 is a three-part webinar series spanning a range of topics on COVID-19. We've already covered PGC's COVID-19 testing and whole genome sequencing during Webinar 1 and Webinar 2. Today, our PGC speakers will talk about bioinformatics analysis as applied to the whole genome sequencing of SARS-CoV-2 or COVID-19. To formally start the webinar series, we would like to invite the Program Director of the Philippine Genome Center in Mindanao, Dr. Larry Annie Murao, to give her opening remarks.
Good morning, everyone. Thank you for joining us today in this series of PGC Talks. We welcome everyone from all parts of the country, all the way from Luzon down to Mindanao. So as you have seen in the earlier video, Philippine Genome Center is really all about making science work for the people. And that includes, of course, addressing our current pandemic. So for this series of talks, we aim to educate everyone about the science behind SARS-CoV-2 testing and sequencing. And we have invited mainly high school students as well as researchers because we believe that you are the future of science. So we would like for you to be able to see how we can make science work for the society so that in the future, you could be able to bring also science into something relevant for the society, especially for omics. Philippine Genome Center is really all about our specialization is all about omics. So today, we're going to talk about bioinformatics. Maybe some of you haven't heard of that word before. But for those of you who have attended the previous sessions, last time we talked about sequencing. But sequencing is only half of the story. Now, how do you make sense of all those information that we get from sequences? That's the job of bioinformatics. So without bioinformatics, we also cannot do anything about all the sequence data that we have. So these people who are going to make a talk today play a very important role also in the field of omics. So I hope that you will be able to get a lot of insights as well as ideas about how bioinformatics goes, and perhaps also inspire some of you to be bioinformaticians in the future. So without further ado, we would like to open the program now for our third session.
Thank you, Dr. Murao. We would also like to acknowledge the members of the PGC Executive Committee, as well as our colleagues from the PGC facilities in Visayas and Mindanao. We are also joined today by Mr. Carlo Lapid, the Senior Bioinfo Specialist of PGC Core Facilities for Bioinformatics. Lastly, here are a few reminders we will be observing during the webinar. For our Q&A, selected questions will be posted on screen after the lectures. This will be answered by our corresponding speaker and our reactors. Questions can be submitted by clicking the Q&A box below. Feedback form, the evaluation form link will be posted in our Zoom chat with specific guidelines on how to answer. This webinar is also being live streamed via the Philippine Genome Center's Facebook page. The link is on the screen. For questions or immediate concerns regarding this webinar, you may reach out via our email address that you can see on the screen. It's capacitydevelopment@pgc.up.edu.ph. Now, let me introduce PGC Mindanao's Bioinformatics University Research Associate, Mr. Paul A. Gaite, who will present his talk on analysis of SARS-CoV-2 genomes, making sense of the genome sequences. Sir Paul, you may now share your screen.
So good morning, everyone. I'm Paul Gaite. I'm the Bioinformatics Research Associate at Philippine Genome Center, Mindanao Satellite Facility. And I'm going to talk about the analysis of SARS-CoV-2 genomes, making sense of the genome sequences. So maybe judging from the title, it's a bit of a serious or technical topic that I'm going to talk about. But I will promise you that I will present it in a more digestible and easy to understand format. So as you will see later, I have a bit of a surprise for my viewers, as I'm aware that most of the viewers are actually high school students. So let's move on first to the world of Pokemon first. So imagine that you're a player in a Pokemon game. So you're familiar with this screen, right? So you're walking around the Pokemon world and you encounter a wild Sandshrew. So a wild Pokemon appears. So, so what happens? So thanks to Professor Oak, as shown here, um, so thanks to this Pokemon professor, this is Professor Oak. So, okay. So this is Professor Oak. So he was the one responsible for creating the Pokédex. So you can liken this to a real world database of sorts. So Professor Oak. So so Professor Oak is a professor or a researcher of Pokemon. So he compiled some information on all the Pokemon that he discovered into this database called the Pokédex. Um, so when you encounter this Sentret, so you have a Pokédex on hand, and this accesses the database on the Pokédex, and then identifies what you have just encountered. So it's a Sentret. So it's a scout Pokemon. So it has a very nervous nature. It stands up high on its tail, so it can scan wide areas. So what I'm talking about here actually is bioinformatics. So basically, bio, so meaning life, and informatics meaning information. So um, basically, what I will be presenting is a practical walkthrough on maybe how you can extract information, as much information as possible from the organisms that you might encounter, as for example, as a Pokemon researcher or as a real world researcher. So, so also in the Pokédex, Professor Oak has also researched on the certain areas where the certain Pokemon is located. So as you can see here, um, so PG is one of the Pokemon, and Professor Oak has determined that the PG is found in these certain areas. However, in the case of Bulbasaur, which is a Pokemon, maybe it's not as well researched as the other Pokemon, so the area is unknown for this Pokemon. So in the Pokemon world, Pokemon are known to evolve. So evolution means that, for example, Sentret, which is a scout Pokemon, its height is 2 feet and its weight is 13.2 pounds. So at a certain level, for example, at level 15, this evolves into a Furret, which is another type of Pokemon. So Furret, as according to the Pokédex, it's 5 feet in height and 70 pounds in weight. So it's a bit longer and a bit heavier than your Sentret. So so that's what you call a Pokemon evolution. So with the evolution, the Pokemon changes form, and some of its characteristics are changed as well. So Ferret has a very slim build. So when under attack, it can pass through narrow spaces. So you can also liken this to the real world, as some organisms quote unquote evolve or change characteristics as time goes by. So in the Pokemon world, some Pokemon vary slightly in terms of their characteristics. Well, not necessarily like Pokemon evolution, as mentioned previously, but they vary in some characteristics, while having most of the characteristics be identical or similar. So you can see here the centret, the previously mentioned centret. The left here is a brown centret, and the one on the right is a yellowish centret. So this is what is known as the shiny Pokemon. Um, so these shiny Pokemon are colored a bit differently from the original one. So do keep this in mind as we go on to this presentation. So this may also apply in the real world as well. So just keep in mind that Pokemon have some sort of slight variance as well as in the real world. So you're walking around again after encountering the Sentret previously, but then you encounter another wild Pokemon. So this is called a missing no. So as Pokemon avid fans of Pokemon might know, missing no is actually a glitch of sorts in the Pokemon game. But then you encounter it anyways. So it's a wild missing no. It appears, another wild Pokemon appears. So Sino, missing no is actually mostly an unknown Pokemon. So maybe Professor Oak hasn't researched much on this Pokemon, so it comes up as an incomplete Pokédex entry. So then you have question marks, but then you can infer that its height is 10 feet tall, and it weighs really heavy. It's 3,500, about 3,500 pounds, and it doesn't have a certain Pokédex entry, so it's really not a very well known Pokemon, or at least to Professor O. Um, so from here, how can we further identify this Pokemon then? So as I've mentioned, so you encountered it face to face physically. So some information are available right there and then, such as the height and weight. So these can be feeds. Also, what are its other physical appearance or characteristics when you first encounter it? So you might want to record it so that it can be recorded accordingly to the Pokédex database. Um, Also, you can, if you're if you're such a fan of Pokemon, or you're really gunning to be a Pokemon researcher, you might have like chemical reagents or tools at hand. So then you can examine further its, for example, its biological or chemical characteristics. So you must be such a geek or a nerd to carry like tools like that. So maybe hypothetically. So you can identify or ID it using these biological and chemical characteristics. Um, Also, you can maybe you have a lab of your own at home. So and maybe in the Pokemon world, these sorts of technologies are available. So you can maybe take a sample of the Pokemon that you encountered, and then, for example, it's a microscope. So you can get a sample of the Pokemon and then look at it under a microscope so that you can maybe take a look further into, for example, at the microscopic level, what this Pokemon looks like. So next, so you might be curious about how this missing no evolves. So maybe this is one of the research publications in the Pokemon world. So it outlines how the missing no evolves. For example, it's actually maybe a Kangaskhan baby, and then at a certain level, it actually evolves into a Kangaskhan, or if the baby's mother dies, it becomes a Cubone. So those sorts of things. Like the sentient that we mentioned earlier, this missing node might have variants, but we are still not sure because this Pokemon is not really well known yet or well researched or researched by Professor Oak himself. So we might be curious about this one. Also, how are they related? So just to give an example from the Pokemon world, maybe these four Pokemon here have a common ancestor. So, and as this tree diagram shows, maybe this from this common ancestor, it gave rise to an offspring that developed a pouch. So then it became, and then it gave rise to a Kangaskhan. Or maybe it developed horns as time went by. Then it's, and more time went by, it became a rock type. So then it became a Rhydon. And then it became a poison type. Then more time came by, and then the toes fused. Then it became either a Nidorino or a Nidorina. With the loss of teeth, this one gave rise to this, and when the toes fused, gave rise to this. So now, remember what I have just presented earlier. And then let's move back to the real world. So what is happening right now is there is a COVID-19 or SARS-CoV-2 pandemic. So basically, a wild virus appears. So maybe liken this to what we have, what I have walked you through with the missing no Pokemon. So this is all over the news, of course. So since late 2019, December 2019, so we have a wild virus called a SARS-CoV-2. So SARS-CoV-2 stands for severe acute respiratory syndrome, coronavirus 2. Since this is the second, the two there indicates that this is the second SARS coronavirus that appeared that is pathogenic. And this gives rise to, when you're infected with this virus, more likely you will have COVID-19 or coronavirus disease 2019. 19 stands for the 2019 that this virus was characterized. So it was first found in Wuhan, China. And it was hypothesized that this virus came from this seafood market, the Wuhan seafood market. They have noticed that some of the residents in Wuhan developed a respiratory illness, and they traced it back to workers working in this seafood market. And reports say that the Wuhan seafood market is known to engage in an underground illegal trade of animals. So, so scientists and doctors were investigating this virus, and so they took samples and they looked at it under the electron microscope and further did some sequencing and bioinformatics analysis. So from those analysis, they found out that it is a SARS, it is a coronavirus, and it causes severe acute respiratory syndrome, as they have observed from the patients. So doctors have found out that yes, it is a respiratory disease. So this is a SARS-CoV-2 virus. It's a respiratory illness. So how does it enter the host? So it, for example, in the air that this woman is breathing around, some of these virus particles are present in droplets, for example. And then the human host, inhales this. It goes through the nose, then through the throat, and then to the lungs. And the virus encounters the respiratory cells in the respiratory system. So the way it does this is the way it encounters these respiratory cells is that you can notice that in the SARS-CoV-2 virus, there are structures such as the spike protein here, this one. Actually, the coronavirus is derived from the shape of this spike protein. And then the virus enters, so the primary point of entry is through these spike proteins. And the spike actually contacts with the ACE2 receptor, which is found in the respiratory cells. So what happens is that as the spike protein contacts these ACE2 receptors, so the membranes from the virus and the cell fuse together, and then the virus is able to enter the cell. So, so further processing, would involve the genetic material of the virus. In the case of coronavirus, this is an RNA or ribonucleic acid. So the genetic material of the RNA gets released. And then this genetic material, this RNA, um, is actually like an instruction or a guidebook. As previous presenters have mentioned, genetic material is the instruction guide um, for the certain organism, or in this case, the virus. So then this genetic material, um, actually what it does is that it instructs the cell's own machinery to produce more of the virus itself. So the different components get gen get produced. So that includes the spike protein and the other parts of the virus, including also the RNA. So these get assembled, and then you then get a full virus viral particle. So enough of these viruses, this would break the cell open. These get released, and then the cycle continues to the other cells in the host. More on the virus, scientists have hypothesized that these viruses originate from bats through sequencing of the virus. So they found that the SARS-CoV-2 virus is genetically similar to those coronaviruses found in bats, and further hypothesizing, they found out that most probably pangolin is intermediate reservoir of these viruses, and then maybe there's like a sort of exchange between pangolin and bat pangolin. Then this gets eventually passed on through the human. So more on that later. So now we gathered those information previously. So maybe as Pokemon researchers or as real life researchers, we're still curious to find out how this SARS-CoV-2 operates further, or how this SARS-CoV-2 infects patient to patient with very efficiently. So then what the scientists did in Wuhan or in China was that they sequenced the virus. More specifically, the RNA, the ribonucleic acid, the genetic material, and they subjected it to next generation sequencing. So this has been actually mentioned by previous speakers. So briefly, I will explain it. I will outline it. So since this is a coronavirus, so this is RNA. So what the scientists originally did was that they converted this RNA to DNA so that it's more stable. Then they did library preparation. So they basically cut up the genomic DNA into smaller fragments, attach adapters so that um, the, s- the sequence segments can be identified from one another um, in the samples, as usually the sequencing runs are pooled. Then these are sequenced by machine. In PGC, usually this is an Illumina platform. And then the sequencer will output short reads. Since it's an Illumina platform, it's a short read sequencing technology. So it outputs short segments or short sequences. Then these are aligned to a reference. So the reference that they used was the SARS-CoV-2 found in Wuhan, China. And then from this, you can gather information such as single nucleotide polymorphisms, or SNPs, indels, structural variations, CNV. But I will discuss more on SNPs later. And I will discuss this process later on in this presentation. So from sequencing the virus, so they found out that the SARS, they found out that the SARS-CoV-2 genome is in this form. So as has also been mentioned by previous speakers, the SARS-CoV-2 genome uh, is, so basically they derive this from assembly or the processing done here, and that it codes for um, the different proteins in the present in the virus. So all over the news, we hear the spike protein. So this is actually located in the 21,000 to 25,000 position of the genome, which is basically as a sequence of nucleotides, which are represented by adenine, A, T, thymine, C, cytosine, and G, guanine. So these four letters. And then, but in the case of RNA, actually, the T is changed to U or uracil. Um, so more on that. So the spike protein gene is, um, so that one is coded by this region. So please keep this in mind as this will be crucial to further in this presentation. So more on how the SARS-CoV-2 virus operates. So the virus, so as I've explained earlier, the virus multiplies in a host and then it gets passed on because it's a respiratory illness. So and it resides mostly in the respiratory system. So the virus is coughed up, for example, by this infected girl or woman. So she passes on, for example, the virus basically from her to a person, most likely without a mask, and then the reproduction cycle of the virus continues into this person. So innate to viruses, including coronaviruses, so what happens is that RNA is changed slightly um, to another form. This is what is called a mutation. So I will explain that shortly. So what happens is that the parental strain is converted to a variant. So there's a slight change in RNA. So I will explain more on mutation here and what this means. So for example, as presented earlier, the spike protein is encoded by this region of the genome. And then, for example, since mutations are more in mutations, mutations are basically mistakes done during the reproduction of the virus or the copying of the RNA, so that, for example, in this case, you have a sequence here, and then an A gets changed to a G. So that happens during mistakes in the copying of the RNA. So what does this mean? So for example, it's all over in the news, the existence of the D614G mutation of a virus, of the SARS-CoV-2 virus. Um, so in the D614 variant of the virus, so you have a GAT here. So this codes for aspartic acid. But then a change in from A to G converts this to a glycine. Maybe just as a review of the Central Dogma. Um, so you have a nucleotide sequence in your genetic material. These are grouped into three. Then each is called a codon. Then each codon codes for a amino acid, and a group of amino acids. A group of amino acids would produce a protein. So in this case, this is the sequence of the spike protein. So a change in from A to G, so from asparagine to glycine, So this one is now changed to the known D614G mutated variant of the virus. So what does this mean? So the change from asparagine to glycine, so it changes somewhat the characteristics of the spike so that the spike protein is able to contact the ACE2 receptor, as previously mentioned, that are present in the cells so that the virus can efficiently infect the cells more. So um, so then they found out that this G614 emerged in Europe. So this was in the around the second to third week of February, and then they noticed that as time went by, this was the more prevalent variant, and they can correlate this to the change in the interaction of the spike protein to the ACE2 receptor. So please keep this in mind as presentation goes by, as this will be um, important to uh, the discussion. So then I've discussed that this mutation, uh, rather as a caution for like it can be the mutation can be seen as a negative. The mutation can change characteristics of the virus so that it can infect the cells more. But then you can also use it to track um, how the virus changes. So for example, as shown in this figure here, so let's say this purple dot represents a parental strain, and the D614G variant can be represented as this, just for example. So then you can monitor the changes in the sequence so that you can track how it changes, and at the same time, with the changes in the sequence between variants, you can track chains of transmission, such as this, for example, in this shown in this map. So just a minor example, for example, from Hong Kong to Australia, um, since you've been able to track changes in the sequence, so you can say that a parental strain came from Hong Kong, then um, you can see changes in the sequence that you can see changes in the sequence so that now it's already in Australia and vice versa. So tracking these mutations is what is called the biosurveillance. So you're able to keep track of changes in the virus or the virus itself. So you can monitor evolution, such as like what I mentioned in the Pokemon world. So you can monitor the evolution in this way. So then, so based on what I've discussed previously, um, scientists were able to sequence the SARS-CoV-2 virus using next generation sequencing. So it outputs the sequence, and then you get all these fragments. Then these fragments um, are have these sequences in fragment form. And then I discussed about the SARS-CoV-2 genome structure, um, the role of mutations in changes in the protein, spike protein in particular, and by surveillance efforts. So then the question is, how do you turn the information that you have? So remember, this is all about bioinformatics, or extracting as much information from your organisms, or in this case, a SARS-CoV-2 virus efficiently. How do you go from extracting all of for example, the genetic material and all these sequences to the useful information that I've presented? So how do you go from here to here? So that is what I will be discussing in the next part of my presentation. So just a brief outline of what I'll be discussing. So I will be discussing about the reference-based assembly and variant calling workflow and phylogenetic analysis of SARS-CoV-2 sequences. So I'll be discussing that in a bit. So remember the previous step that I've discussed. So you have your genomic DNA. This was fragmented, then sequenced. Then you yield all these short fragments. This gets aligned to a reference, yielding information such as Snips or single nucleotide polymorphisms in those structural variations are in CNV. So briefly, um, so during the before like sorry at this stage, for example, so this corresponds to this step. Um, in briefly, so these are actual sequences, for example, CATE and this fragment here and this fragment here. So what assembly does basically does is piece together these fragments, and the software looks for sequences such as this on the ends. So for example, in this case, GTTA. So the assembler would look for a sequence fragment containing this sequence, and then it intuitively it pieces this fragment sequence fragment here and here, and CATE. Then it looks for other fragments that are containing this one. Then it pieces them together, all pieces them together to form this assembled sequence. So in a nutshell, that's what will happen in the method that I'll be discussing further. Um, So the reference-based assembly and variant calling workflow is as follows. So you have read mapping against reference Sarsko V2 genome sequence. So this will be done by BWA SOM tools, primer soft clipping and quality trimming by IVAR and SAM tools, initial variant calling SAM tools IVAR, removal of reads with mismatches as primary regions IVAR, consensus assembly generation by SAM tools and IVAR, and finally variant calling for consensus assembly, which is performed by RAT and Mummer. So what happens with read mapping against reference SARS-CoV-2 genome sequence? So the Illumina platform produces a set of short sequence reads. So what the software does is it maps it. Basically, it matches the sequences to different regions of the reference genome. So such as this. So you get this sequence. So you have this reference sequence, and basically it aligns the short sequence reads to different regions. So for example, in this case, TAA's GCG, it might have a mismatch because how it maps, it depends on the parameters that you enter into the running of the software. So it matches it even though there are some mismatches. And accordingly, it also aligns these sequencing reads here and here, although albeit with some mismatches. Next is the primer soft clipping and quality trimming. So how does this work? So as I mentioned earlier, when you use the Illumina platform, you use primers and you attach, sorry, I mean, you use sequencing primers so that so as to amplify your reads so that you have more reads of available, and then you can simultaneously sequence them so that you'll have more accurate results. So that primary sequence gets uh, basically it gets included in the uh, your sequencing reads. Then you also attach sequencing adapters. So what primer soft clipping does is that it basically is a quality control step to remove, or maybe um, at this step it basically like alerts the software so to say that in this certain region you have these primer regions. So that in more downstream processes, the primer regions get removed. quality trimming, because in Illumina sequencing technology, you have what is called the phasing. So this lowers the quality of bases at the ends of the sequencing reads. So then the quality trimming removes these so that you will have more accurate results downstream. Next is initial variant calling. So what this basically does is that so you get all your aligned reads together, mapped to the reference genome, and then the software looks for on a base per base level differences um, on the base calls with your reference genome sequence. So in this case, you have an A in your reference genome, but then when aligning all those reads together, you get a G instead. So this gets tagged by the software, and it calls it as a single nucleotide polymorphism. Basically, this indicates a single base difference from your original reference. So A in the reference genome sequence, it's called instead as a G, at least in this data. Next is the removal of reads with mismatches at primary region. So I'm showing the figure that I showed previously. As you can notice, for example, in this read, you have these sequences at the end. Uh, you can say that this might correspond to a primer region. So as I mentioned earlier, so primer sequences get integrated into your actual sequencing read. But actually, these are not um, actual from the actual viral sequence, but these are because these are integrated because of your experimental protocol and not because of these are from the actual virus itself. So what you do is that so you have these primary regions, but then it because sometimes you have errors in the primer primer sequences. For example, the intended primer did not attach to um, the sequence of interest. So there's a mismatch. So what you do is you remove, for example, this one is a C. For example, this will, if there's a C here instead of the T in the original intended primer sequence, so you basically remove these because these are erroneous. So then you get to remove erroneous reads. So then you'll have more accurate reads at the end. Then consensus assembly generation. So so you have all these reads mapped and initial variant calling. Then you then get to generate a consensus sequence. So for example, in this case, you have an A in your reference sequence, but then you have T base calls in almost all of the reads except for this one. But then the definition of consensus is that you rely on what is most observed, what is the base that is most observed. So it's T actually. So then the consensus sequence is actually T instead of the A. Then you have a C in this case. Um, this is the majority of the base calls, but then you have a T here, but then of course it's a consensus, so you have a C. So you generate the consensus sequence that way. And lastly, you have variant calling for consensus assembly. So a variant calling is once again done by RAT and Mummer, especially RAT, which is the Rapid Annotation Transfer Tool. So it employs algorithms that incorporates, for example, sequencing errors such as this. In this case, there's a majority C call, but in one read there's an A. Or in this case, there's a legitimate snip call because many of the reads contain A instead of your reference G. So the RAT tool relies on this and uses its models or algorithms so that it takes that into consideration so as to generate a more accurate consensus sequence assembly. So then, so previously, so we have generated complete genome sequences of your SARS-CoV-2 from consensus assembly method. Now you're ready to do phylogenetic analysis of these SARS-CoV-2 sequences. Now, outline of this method. So you have multiple sequence alignment of the SARS-CoV-2 genomes by MAF, automated trimming of aligned sequences by TREMAL, evolutionary placement of newly obtained sequences by RAXML, and plotting of the phylogenetic tree by ggtree. So let's look at a brief overview or short introduction of phylogenetics. So as I've mentioned earlier, so you can track the origins of a certain variant from, for example, a certain variant that has arisen. You can track its history or how it evolved from that variant to from the parental strain that it originated from. You can do that by employing phylogenetics, which is basically a study of the evolutionary history of organisms. So, or in this case, a virus. So, for example, you have virus variants A, B, C, D, E here. Um, Intuitively, from this diagram, you can say that, for example, virus variant A and B are related to each other through this common ancestor. C and D are related, for example, to A and B through this ancestor here. So they um, branched away from this common point here. And E is very distantly related, for example, to A, B, C, and D through this ancestor. So this is basically what phylogenetics does. And so, and also in the horizontal scale, you have from ancestors to your present day species. So, so as I mentioned earlier, virus variants arise because there is a slight change in the sequence. So then, theoretically, you can track them down. You can track the phylogenetic since as time goes by, for example, there's what is called a molecular clock, wherein as time goes by, since virus naturally mutate, so basically there are base changes as time goes by. So you can track, for example, the evolutionary history or how the variants arisen through changes in the sequence. So for example, you have a multiple sequence alignment of these virus samples here. And you can track how these changed through this diagram. So as follows. Um, and then you can infer relationships that, for example, this virus is related to this virus variant somehow, closely related, and also this one to this one. Then those two are more distantly related through this ancestor. So then you can infer that with actual SARS-CoV-2 virus variant sequences derived from next generation sequencing of the SARS-CoV-2 virus. So how does the phylogenetic analysis come through? So you first do a multiple sequence alignment of SARS-CoV-2 genomes. So as I've shown earlier, so this is an example of an alignment of SARS-CoV-2 genomes. So algorithms that are actually different from the ones used in consensus assembly generation are used. So these are aligned. So whole length genomes are aligned together. Then you do automated trimming of these aligned sequences using TREMAL. Since the algorithms cannot really deal with these differences in the lengths basically these sequences here. So you have to trim them using Tremile. And then afterwards, you do evolutionary placement of newly obtained sequences, assuming that existing sequences are present. So from the same figure that I've shown before, um, inferring the phylogenetics of the different virus variants. So then if you have a new sequence such as ACT, to added to the multiple sequence alignment. So by the algorithm, you can determine that, for example, this one is related, more closely related to this set of sequences than to these sequences. But they're bottom line, they're all related to each other to these sequences. Lastly, you do a plotting of the phylogenetic tree, which will be discussed further by the next speaker, Dr. Yap. So so this is another example of a phylogenetic tree, and this was generated by GG3. So then you can use GG3 to make your phylogenetic trees look much more presentable or publication worthy quality. So to wrap up everything, so I've just presented a basic like practical walkthrough on how real world scientists might encounter a new virus such as SARS-CoV-2, and then how to further analyze, for example, through its sequence, and gather um, more more useful information from that. So I'll pass this on to the next speaker, Dr. Jan Yap, to for the next part of the presentation. So thank you very much.
Thank you, Sir Paul. As you mentioned, we will now proceed to the next speaker for the webinar. Let me introduce PGC's core facility for Bioinformatics Program Director, Dr. John Michael Yap, who will present his talk on BioArt, presenting learnings of sequences for make benefit glorious discipline of bioinformatics. Dr. John, you may now share your screen.
All right. Thank you very much, Colette, and thank you very much, Paul, for that extensive walkthrough of the bioinformatics pipeline for biosurveillance. Okay. So, good morning, everyone. Kaya pa ba? Di pa yan? Yeah. So, the title of my talk is By Art, Presenting Learning of Sequences for Make Benefit Glorious Discipline of Bioinformatics. Okay. If you didn't get the reference, it's, it's all right. It's okay. Okay. So, before I truly begin, okay, one of the things that uh, I would say I hate about the pandemic was that it took away the, our capacity to come together to converge physically and conduct a physical, space, uh, physical congregation, have this forum in a, actual venue. Okay? Because talking like this somehow takes away interaction. And just like Borat, right? It was his films, although taste is somewhat questionable, the idea of interaction, okay, to be able to elicit reactions from the audience and to bring about the what you call the message is what I would have wanted in something like a forum, like in something like this public forum. But nonetheless, allow me to at least stimulate that kind of thing. Okay. So content warning, possible jump scares. Next slide. Let's first play a game. Okay. So it's just a simple game. No, nothing fancy. Nothing fancy. So a picture will be flashed for 3 seconds. Okay. Afterwards, a question, right, will be shown. Type in your answer. Type your answer in the chat area. All right. Okay. So ready? Okay. Okay. I'll count up to three just to give you the timer. Okay. Here's the first pick. Ready? Here we go. One, two, three. Okay, so audience, what was that picture all about? So it was a lemur. Okay, lemurs. Yeah. Okay. What about the lemurs? Can you give a brief description or at least concoct a story about the lemurs? No one. Okay. Cooperation? Yeah, in a sense, cooperation. There's some sort of cooperation. In fact, they're sleeping together. It's a cold day. They're trying to get, keep themselves warm, right? Right. So, recap on the rules. I just showed you three picture for 3 seconds, and then just tell me the story. Tell me what was that all about. That's all. Just type it. Okay. So, basically, okay, it's a conspiracy. Okay, that apparently is the what they call this, the aggregate um, noun to describe a group of lemurs. It's a conspiracy of lemurs sleeping together, trying to preserve one. Okay, okay. At least you got the idea, right? All right. So I have a second picture. All right. Again, I'll count to three. Okay. Once the picture is shown, I'll count to three, and then I'll I'll move on to the next slide. I'll ask you what was that all about. Okay. Ready? Okay. Ready. Here we go. One, two, three. Okay. What was the picture about? It's an alignment of what? Okay. So multiple sequence alignment. So that said, props to what do you call this? Props to Paulo, Paul Gaite for making sure that's imprinted on it. DNA, yes, because it's easy. But what about it? All right. So, right. So basically, you just got the gist, basically, right? But you did not know the story about it. At least not as good as the lemurs picture, right? Okay. Now, in this day and age of Industry 4.0, where data is the new oil, it is also a new soil to be able to come up with different information and knowledge, pieces of knowledge that could help humanity, right? And bioinformatics is no different, or the field of genomics is no different. You try to infer meaningful information that could help out um, um, different types of people, from epidemiologists to other scientists, and even people make sense of the biological data that they have. Okay. Now, the idea is, um, though, is that sometimes it the information is not so apparent. Okay, unless you present a visual representation of the information that was presented. Right. A few, wise men did say that, a picture paints 1,000 words, and it is the same with bioinformatics. Now, as Paul has mentioned in his lecture, right? Um, by and large, what we're gunning for the biosurveillance, at least, is to identify variants that are present here in, the country, right? And we call this. And we do this through bioinformatics analysis, through variant calling, variant analysis, and also through phylogenetic analysis to see possible similarities and possible clustering, thereby leading to um, identification of variants, be it existing ones or possibly new ones, right? And I think you've heard of this before, right? Phylogenetic tree uh, visualization, okay, which is in this case a radial tree, right? It's the radiality here is more of for presentation and for space saving, right? Now, in phylogenetic analysis, I think Paul already mentioned that the idea here is to try to find an evolutionary family tree for the viruses, in which case here's the SARS-CoV-2, partial phylogenetic tree, right? Now, apart from just generating the tree, we could put in a couple of colors to show the clusters that are, present or that are seen here to make a more visual, what they call this, representation of the information that we inferred from the analysis that we did, right? In this case, we used color, right, to represent the linkages. Sorry, not the linkages, but the variants. All right, or the lineages, not linkages, lineages that are available. So you have here the yellow um, ones. I think representing the. Uh, Let me check my código. Okay. The yellow ones represent the B1128, and which is a variant discovered in Brazil. You also have their orange P1 from Brazil. And then you'll notice here the purple one, the B117, which is also the, what do you call this, the UK the one discovered in the UK. And then you have here the red one, which seems to come out of, or is, or sorry about the low res picture here, seems to be to have evolved or to have come from an orange or yellow. Um, Actually, it's a yellow one lineage, okay, which is the what we now know as P3. Okay. Here we see that the P3 actually came out of B1128, something to that effect, or has at least has an evolutionary relatedness to it, right? As per analysis. And then furthermore, we could also or more or less estimate the time from when these certain lineages came about. As seen here, wherein you have concentric circles representing months from September 2019 to December 2020, representing the lineages, or sorry, the time. Okay, so if you would see here, P3 might have come somewhere when it might have uh, evolved into existence, so to speak, around September, around that time, September 2020, right? So phylogenetic trees, when visualized properly, not only gives you information on the clustering, it also gives you the information of the relatedness, evolutionary relatedness of the um, of certain variants, and also it could also possibly give you an estimate on when they came into existence, right? Now, uh, that's for phylogenetic analysis. Now we also have, something for variant analysis. Okay, so basically this is almost a simple bar plot, right? Basically, here the heights of each bar plot give you the number of samples. Sorry, first things first. Okay, here the x-axis more or less gives you the position, the genomic position, that uh, your SARS-CoV-2 genome, right? And then each here positions here are what we call this not Snips, but let's just say gene loci of interest, okay, where mutations were observed to frequently occur, right? And then here the bar charts represent the number of samples in the analysis that okay, not necessarily we, but the team conducted to be able to come up the what they call this to in the analysis. Okay, so here the main idea in this one is that this is to profile the mutations or the signature mutations of the emerging back then the emerging P3 variant. Okay, here we identified that in the samples that we analyzed, okay, none of them seem to what we call this align or match the signature mutations of the existing variants then P1, B117, and P1.38, the one from South Africa. Okay, that's why if you notice here below, you have three what they call this arrays, okay, representing each of the known variants, and then you'll notice that some of the boxes are colored, okay, representing the signature mutations for each one. So you'll notice here that none of the mutations observed in the samples perfectly align with all of the signature mutations of the variants of concern uh, that were flagged. Okay, so here visualization also helps um, characterize the variations or that are present in samples, thereby possibly um, discovering or detecting new variants that otherwise not been detected prior, right? And then, of course, I think we've seen it before. We also do simple you know, visualization using a choropleth map, okay, wherein we pinpoint the distribution of the um, lineages across the areas where the sources or these specimens were sampled. Okay, so pretty much basic stuff. So we even we don't resort sometimes to very complicated visualizations. We try to make it simple so as to make it a little bit more understandable, more relatable to other people, especially the layman. Okay. But here's the question: we do have visualizations, but pictures enough to give picture. Okay. What I'll be showing are some pictures, okay, or screenshots, okay, not necessarily of bioinformatics related. Okay. So this is an April 4 or April 5, we call this infographic, part of an infographic released by the Department of Health regarding um, ICU bed utilization and ventilator utilization. So focus on the ICU beds, right? So it says here 79% utilized. Okay. So basically, yeah. Okay. Good. So at least we'll say there's still 21% that are unutilized. Correct? Okay, good. But if we take it somewhere, okay, we zoom in. I took this from a rheumatologist from Manahan. Okay, here's the contextualization of some of at least to where he is currently serving in as resident doctor. It's reported that the COVID ICU was 92% full, but guess how many beds were vacant at that time? One, right? Their ICU, at least their hospital only has 12 beds. So don't take that vacant 8% amounts to that many, at least in that context. So here, we still need the help of, communication arts, okay, not only of the visual arts, to be able to convey our, the information that we want to the people targeted audience to partake, right? So letters still matter, right? That's why we still publish, okay, technical reports or scholastic papers so that other scientists could have an appreciation or, have a critical analysis of the work that we did and the results that came out of it, right? We also have um, we also, well, at least our boss, executive director, Dr. Cynthia Saloma, um, you know, um, takes interviews, okay, from different media outlets because we still know the power mass media still has to be able to propagate timely and correct information regarding what we're trying to do. Also, like why we're here, okay, we also conduct public fora to be able to reach out to as many people as we can using online platforms, okay, and also to identify demographics that could be, um, you know, a good recipient of the knowledge regarding these matters that come out of our work. Okay, and then, and sometimes, okay, especially, and this is especially true for the specialists that we work with, we also conduct lectures, right? So to be able to guide them on how to assess or appreciate the work that we do properly. So that basically is how visual and communication arts help bioinformatics, and especially how it is being done here in our biosurveillance. So that technically ends my report. And oh, wait. Oh, hold up. What was that picture all about anyway? By that, this one. Okay. So given what we've learned, here is what is it all about? Okay. What you see here are the first 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. The first 15 full genome Sarsko V2 Genome sequences that were submitted to GenBank, okay, earlier in the stages of the pandemic, right? And what I wanted to show, or the story there, is that at this point, you know, the differences between the genomes, all right, save probably for one, which is the reference, okay, isn't too large yet as compared to how it is right now. So that was the story behind the picture that I showed you a while ago, right? Okay, so now since this is also some sort of movie, let's go to credits. Okay, I want to give credit where credit's due. So we have here the core facility for bioinformatics, core team members. So we have um, our lead um, bioinformatics specialist for this biosurveillance effort, Francis here, ably supported by Mr. Carlo Lapid, okay, who does the haplotype and the phylogenetic, partial phylogenetic analysis. Okay, we also have El King Morado, our communication specialist and the one who does the reports and parts of the visual. And then we have Mr. Joshua Dees and Ariel Gabriel, who make sure that the servers are up and running when we have to do the analysis. Right. Also, apart from the core team members, we have the bioinformatics biosurveillance team. All right, who is headed by our resident K-pop boy band member, Mr. Kenneth Kim. Okay, and who does a lot of the things that are the legwork that is being done, and they're ably supported by the twins, Miss Maria Sofia Ria Yangson and Rihanna Patricia Ria Cruz, right? Who does the informatics part, the information, who builds the information system for this effort. And then we also have Mark Jerome Cruz, our head visualization guy, who is sadly no longer with us. I mean, he's still alive, but he's just out there somewhere, fulfilling his dream of being a Pokemon master. No, really, seriously, as a data scientist. Okay, and then the new tandem, okay, um, uh, who are the new ones who came in. So we have Mr. Aaron Pangilinan and Mr. Renato Jacinto Mantaring, okay, our former interns who have, recklessly way of biosurveillance, and thankfully so, because we need a lot more bodies. And with that, my presentation has truly ended. Thank you very much. And I will now probably turn it over to Atishel. Sharat?
Thank you so much, Dr. Zan. I think a lot of our viewers right now would like to thank you for your very informative and very interesting way of presenting your data. But before we go through our Q&A section, let's just open a discussion about what has been presented so far. So let me invite our guest reactor, and I mentioned that he's also the senior bioinfo specialist. That's part of your group. We would like to know more about his experiences in sequencing the SARS-CoV-2, or basically what was his experience while he's part of the team. So, Sir Carlo, what are your thoughts about the initiative that we have today? I-share yung pag-sequence at yung pag-training mo sa mga tao sa Mindanao.
Hi, Shadet. First, thanks for having me here. No, that's a very interesting question that I was not entirely prepared to answer because it's been such an eventful past 12 or so, magsa 13 months na tayo, no, on various stages of lockdown. Uh, of course, there's the shared global experience of having to restrict our lives, staying at home, not being able to go out and see friends and family, which has been very difficult. But it's also been a very, if I'm being honest, both challenging and intellectually and morally fulfilling experience for me. Because it feels good to be able to work on something that uh, is of direct parang relevance and benefit to the country and the world. Wow, that sounds so self-aggrandizing. Pero I guess that's my honest response na the things that Dr. Yap and Polo were talking about analyzing all of these sequences, and Polo was parang talking about yung database nga, yung na-analogize nito the Pokédex. We're the ones, as bioinformaticians, we're the ones that work to assemble and curate and submit that data, both our local databases and the global databases, which can be used by other scientists in our global effort to control this pandemic, we also submit our findings to the Philippine government, which can inform their public health policies. So I think that's the main parang reaction or feeling that I have towards the work that we've been doing these last many months, in spite of all the challenges and difficulties, and there really have been a lot of of difficulties and struggles. I keep returning to how how fulfilling it can be in believing in the importance of the work that we're doing. So siguro that's my main answer to that question, Jeanette.
Thank you so
Ay,
much,
sorry,
Sir
at limit
Carlo.
10.
It's a very human answer, no? Kasi parang kanina puro patterns yung nakikita natin, pero
Mm.
we would want to know how it feels to look at those sequences day in and day out. Parang gusto rin natin malaman ng mga tao na kung nag-run kayo ng sequences, gaano katagal yung pag-run ng sequences? May tulugan ba to?
Well, the work comes in waves, no?
Mm-mm.
Of course, it's a collaboration. Hindi lang naman kami yung as Polo talked about, because the lab work is not done by us bioinformaticians. So in many respects, sila yung minsan all night or weekend nagtatrabaho to do the sequencing runs. And then pinapasa nila sa amin. And then the pressure is on us to minsan nga, true, minsan working overnight or on weekends to parang process that data so we can submit it to the people that need it next, like the Department of Health, for example, or pag kailangan ng masubmit to online databases. So hindi naman sa walang tulugan kasi like I said, it comes in waves, but there are definitely times when okay, all hands on deck. Kailangan nating matabaho to hanggang matapos na. So yeah, those are some of the challenges that I was alluding to earlier. Thanks for for
O,
giving
diba?
me a chance to parang to bring that up now.
Okay, thank you, Sir Carlo. I would also like to call on Dr. Murao, no? Kasi I know that they have a project in Mindanao concerning biosurveillance din, and they're also running analytics. Pero iba yung konteksto nung sa kanila. We would like to invite her to talk more about that and how yung experiences nila sa Mindanao, how it relates to what we're doing as a nation was part of PGC.
Yes, so we did some sequencing here in PGC Mindanao. This is actually in collaboration with Accessible Genomics. I just want to acknowledge them. Accessible Genomics, which was funded by Jogel, and they were able to deploy mean ion sequencing. So these are like portable sequencing units here in Mindanao. And our pilot work focused on wastewater samples because work in other countries have shown that we can actually conduct surveillance of COVID-19 or SARS-CoV-2 also in wastewater because that's where everything, all our wastes end up. So the virus can also end up in our wastewater, and it could serve as like a community level of surveillance, not individualized. And so, yeah, we did some sequencing on our wastewater here in Davao City. We have some preliminary data. We're just working on the data right now to be able to come up with something sensible out of the information that we got. But it's very challenging. I tell you, compared to clinical samples, wastewater is very challenging to work with because naturally it's wastewater, so things can get degraded in the wastewater and your virus may no longer be intact. So that's what we experience from working with wastewater sequencing. So it's kind of new challenge for us compared to clinical samples, and we hope we could tweak our way to improve that system also so that we can also get really relevant information also about the community just by looking at wastewater. It's very relative to clinical surveillance, it's easier because you just go there to the wastewater and collect samples, and you can already get some relevant information. But yeah, the challenge there is how to make it um, accurate, reliable, as well as how to deal with the challenges of all the environmental factors that come into play in the wastewater.
Very interesting yung shinare mo, ma'am, no? Wastewater. So, I guess, I ushered in that concept dahil as people are more aware of how COVID works, pangatlong webinar na natin to, gusto natin talagang ma-highlight na ang daming approaches pagdating sa science. And it's really interesting how fast it's being developed now. Sad din kasi nade-develop siya ng mabilis dahil pandemic nga at maraming apektado. But I'm glad that there are a lot of agencies helping PGC sa Maine, sa Visayas, sa Mindanao. And we are always open for collaborations. So, maraming salamat, Ma'am Lar, Sir Carlo. We will now proceed to some of our questions that that will be addressed by our panelists. So we have a question for Sir Paul. Sir Paul, what is the sufficient depth of coverage for variant calling? How do you verify the Snips found and make sure that they were not generated due to errors only? Are they specific criteria or minimum requirement in the data that you strictly follow?
Based on the literature that I've read, they usually allude to, for example, 20 times coverage for a certain base. So, for example, you have in your reference genome a T, then it was observed that at least for that certain base, you observed A 20 times. So judging on the literature that I've searched, so 20x coverage or 20 times that different base is observed at that certain base would be sufficient to say that this is actually a SNP. And also, I think at least some literature say 5%, some say 10%. So that might also be a metric for that.
Yeah, if I can just chime in our pipeline naman that we use at CFB, uh, 10x coverage is what we use for variant calling to determine SNPs. And then when it comes to detecting errors, like sequencing errors, that's really Paulo touched on this no, in his lecture. That's really an inherent challenge in variant calling. But fortunately, the tools that we use already help with that. They're embedded statistical models that help to distinguish between true variants and sequencing errors. The main difference being that sequencing errors tend to be parang rare or isolated, and true variants appear with higher coverage when you look at the alignment of the reads. So thanks.
Next question. Thank you for that, Sir Carlo. For the other panelists, kung sino po ang gustong sumagot nito. Meron pong tanong dito. Since the SARS-CoV-2 virus mutates very fast and now has formed different variants, are the vaccines made from earlier variants still effective? I'm pretty sure hindi pa rin natin specialty ba ng mga nasa bioinfo yung
Yeah,
information about vaccine?
I can comment on this, but the first thing I'd say is that you're correct. No, none of us are qualified experts to speak on parang vaccine efficacy. That is really a question for the epidemiologists, the doctors, the immunologists. Anything I say will just be a repetition of what I've read in the news, and still the scientists are still trying to answer these questions. Parang I hesitate to. I might I might be spreading misinformation if I comment with any confidence. But I can say that it's a bit of a misconception to say that the virus is evolving very rapidly. Of course, rapid is relative, but relative to other viruses like influenza, actually, SARS-CoV-2 is evolving at a fairly slow or moderate rate. Actually, what we tend to say in analyzing this is that there's relatively low genomic diversity in the global SARS-CoV-2 samples, which has its own advantages and uh, difficulties in analyzing it. But the good thing is because it doesn't evolve that fast, relatively speaking, it gives us hope for keeping up with our vaccine design strategies. That's what I feel safe saying based on my own experience and expertise, siguro.
Yeah, can I add something to that?
Yes, please.
Yeah. So, uh, viral mutations, these are inevitable. We cannot stop viruses from mutating. That's part of nature. It's part of the balance between nature and pathogens. No, they have to evolve also. That's part of their life, kumbaga. So we cannot stop them from mutating. So uh, there will really come a point that these viruses will develop resistance to drugs, will develop escape mechanisms to vaccines. We don't know when that point will come. But one thing is for sure, we cannot stop the virus from mutating, but we can do something about ourselves, you know, from in order to manage this type of possibility. So what do I mean? That means that ang sikreto dito, ang solusyon dito nasa ating kamay. For example, when the government says social distancing, we have to maintain social distancing because that's the only way we can stop the virus from spreading. The virus will only continue to propagate itself if it has a human host. So the more that we separate ourselves from each other, then the lesser are the chances that the virus will find a new host. So yun yung isang sikreto doon. Another thing is with the vaccination campaign that is ongoing right now. Maybe some of us still have some issues about vaccines, but I think it's really important that we call it upon ourselves to be responsible enough to have ourselves vaccinated if we have that opportunity. No, pag dumating na yung opportunity na yun. Have ourselves vaccinated because as soon as we vaccinate ourselves, then that is also our chance to prevent the virus again from spreading. So ito yung mga um, uh, things, that are important also in considering how to manage the virus. The secret really lies in our hands. So we cannot do anything about the virus, but we can do something about ourselves.
Thank you, Ma'am Lair. We have a question for Sir Jan. What sequencing platforms are you using to generate data? And again, the data that has been generated, what comes after that? Paano nagiging accessible sa public yun?
Ah, paano nagiging accessible to public yun? Okay. So first, Carla actually has a better appreciation of this one. Pero I think we're using a NovaSeq platform to generate that. Tama nga ba, Carla? We use NovaSeq or,
Yes.
yes, di ba? NovaSeq siya.
For
The
some of
idea,
the runs we used MySeq, but I think the vast majority of our sequences, I believe we use NovaSeq.
NovaSeq, because particularly high throughput siya, and we could
Mm.
sequence more samples than that, than other usual platforms. Akala namin. The second question was, I believe, was how do I get the sequences? Tama ba? Public? Tama ba?
Public,
Shell?
yeah.
Yeah. Um, okay. For at least for the assemblies, we submitted to GISAID, GISAID. It's actually a global tawag dito, platform for genomic epidemiology of influenza viruses. It wasn't mainly for SARS-CoV-2, but it was eventually parang populated na rin with that one. So doon kami nagsasubmit. particularly because, well, ease ng ng submission relative, as well as yung ano na rin, aptness, yung more or less appropriateness. We could actually submit to GenBank pero medyo mixed na rin siya, mixed bag of sequences na rin. So at least dito sa GSAID mas focus. Ang ano nga lang is that sa GSAID, I think you have to make an account to be able to access it. Hindi siya yung tipong yung Googleable tapos i-download mo siya. Yun lang naman. Pero pretty much open pa rin naman siya na platform.
Okay, thank you so much, Sir Jan. There's an interesting question here. I know that PGC validated the presence of the P3 variant tayo yung pinadala ng sample. Tama, sir, di ba? From Visayas.
Yes, that's correct.
Yeah.
Pero, sino yung mga group of scientists na inaaral ngayon yung properties ng P3 variant para masabi sila na mas potent ba to, kakaiba ba to sa ibang variants? May studies na bang ginagawa about it within the Philippines? Kasi alam ko marami ng studies outside of the Philippines.
A good question actually. May partial na diba, characterization na ginawa si Dr. Neil Basco. Yun dun sa proteomic level analysis, protein level analysis nung P3. I think meron ng preprint na available sa medRxiv. So that's at least that's who I'm aware of. Unless there's somebody else, Carlo, Paul.
Well, we did. I don't know how much is okay for me to disclose now. I think it's okay for me to say na there are efforts underway in a collaboration with PGC, NIH, NIMBB, to put together a group to analyze the P.3 virus. Actually, they already requested some of the sequence data from us. But from the last I've heard this is still in the proposal and funding stages because unfortunately, the gears of our bureaucracy have to move at whatever pace they can manage. I'm not up to date on the latest of how far they've advanced the project already. Lalo na kasi there's some laboratory work involved in parang actually synthesizing and analyzing not the virus itself. They don't actually synthesize a dangerous virus, pero parang parts of it. So that that's not just something you can just start up na basta. That's one of the nice things about bioinformatics. If you already have the computers and software, once you get the data, you can get started. In lab work, you need the reagents, the equipment, and so on, the samples. So there are more challenges getting things started there. Yeah.
Thank you, Sir Carlo. So from what I heard, marami namang interested parties na naggagawa na ng proposal for knowing more about the P3. But my point, parang we just also need to be cognizant na ang dami nating minomonitor, lalo na yung hindi lahat ng tao may skills and competencies sa pag-analyze ng data. Tapos maraming data available at iba-iba yung gusto nating malaman about that data. So I guess what I'm saying here is nakapila lahat. We would want it to be moving faster pag marami tayong resources. Kaya ginagawa nating itong webinar na to para mag-engganyo ng mga tao na siguro kung long-term view yung titingnan natin, sana dumami pa yung mga scientists interested in learning more about this, doing experiments about this. Because honestly, ma'am, sirs, sa capacity building projects natin, ilan po talaga yung nakatapak na ng lab at kayang gawin ito sa buong Pilipinas? Masasabi, are we confident na nasa more than 100 people na yung kayang gawin to? Yung mga ginagawa nyo sa bioinformatics?
Are you?
Hada ko sa question.
That
Okay.
was the
Yeah.
question.
Are
For
you
comments.
How,
specifically
how far are
referring
we
to
how far are we from
bioinformatics?
the goal of
Ah,
Uh,
developing the scientist?
Mhm.
mm.
Actually,
That
yan ang kailangan natin kasi I see bioinformatics as, as part of data analytics, and right now data is really gold. And for this pandemic, for example, bioinformatics has played a very important role. Parang sila yung silent heroes ng ating pandemic. Kayo yung mga silent heroes ng ating pandemic. Bakit? First of all, how did we know that it was a SARS-CoV-2? It was through sequencing, di ba? And through phylogenetic analysis, na-figure out natin, kamag-anak pala ni SARS. So if had we not known kung anong virus ito, I mean, we would be fighting this pandemic blindly. Another thing, vaccines, medications, how were they developed? That's because there were already prior work on this, on sequencing, on protein work. So itong mga work na ito, these were done in collaboration with bioinformaticians. So I think importante talaga ang role ng bioinformaticians. But in the country, parang it's still an emerging, di ba? Parang it's still an emerging field.
yeah.
So like you guys, you're all young because this is a very new field. And so,
ma'am, sila lang.
sila lang. Kasama ka dyan, Dr. Jan. Oo, so kailangan natin, siguro yun yung call natin for our guest audience dito, yung mga high school students dyan, yung mga budding researchers natin dyan. Sino po sa inyong gustong engage into the field of bioinformatics? We highly encourage you to be part of this a journey or adventure, no? Kasi we need more people like these guys, Carlo and John.
Oo nga
And
naman.
Polo.
And Paul, of course, yes.
Yes, I'm actually a
Produkto ng
product
trainee yan
of
si Sir
PGC
Paul.
from
Uh,
PGC. And
Yeah.
I'm
yeah,
still
we actually magaling tinanong mo kung umabot ng 100 na, Shilet, because we actually the last, well, the year before last, because last year was all lockdown. We had a year long parent training internship program where we trained over 100 people in bioinformatics, and like Paula said, he was one of our trainees. So we're moving forward there. We want to really capacitate more people. Sana we had more resources rin to teach and train in addition to all the research that we're doing. But hopefully, the more people we train, they can train people in turn in their own facilities and institutions. So it's an ongoing journey. I don't think we're ever gonna reach. You asked, parang, when are we gonna reach that goal? I think that goal is like a horizon. You never reach there, but hopefully we keep moving forward and grow in that capacity.
And to a certain extent, that goal is also organic. Kasi nag-evolve rin eh, di ba?
Uh,
Right now, ito yung ano
exactly.
natin eh, to be able to do basic na bioinformatics, at least yun lang, di ba? But later on, of course, once everybody can do, not siguro everybody, but a lot of people can do, basic bioinformatics analysis. Now, you will have to advance to the next step. What else can be done, di ba? Ganun naman palagi. And it's not only for bioinformatics per se, no? It's also for science in general. You try to push yung the horizons of what can be done, of course within ethical limits pa rin. We'll have to that. And also, hopefully it should be, as na mention nga ni Shelet kanina, 'di ba, nung sa unang-una pa, it should be in the service of the people talaga. Um, you know, I okay, ako person, this is not a diss track to theoretical people out there, pero yun nga, meron rin naman silang meron rin naman importance yung pag-aaral ng theory, di ba? Pero at some point kasi, you'll have to look for something that is practicable, especially given the conditions uh, currently, di ba? So sana, yun nga, sana eventually we could, for those who are engaged in theory, yun nga, eventually we could bridge that into practice. Of course, hindi natin alam kung kailan yan. Pero I guess the most important thing that I wanted to say is that, yun nga, regardless of where you are in this sciences, you should, even if you're more on the theory part or the more the applied or practice part, talaga, as much as possible, you should keep in mind who you're doing the science for. And it should be for the people.
Sir John, thank you. So, medyo malapit na mag-end yung session. Let me just ask this question, no? And I guess may mga hindi masasagot na questions, but this opportunity would help people know more about bioinformatics. Na-mention natin na you have been doing training for bioinformatics. And iba-iba yung audience. May pang high school, may pang working professionals na. What are the plans for that kind of training itong 2021? Meron po ba tayong mga balak na gano'n?
Mm.
Kasi parang, as mentioned by Doc Lair, parang we can always swab and process the material. Pero pag hindi naman natin na-analyze, it would be meaningless. So people should know more about putting meaning into what we're doing. Do you have any plans? Does your team have any plans to expand
Yes.
your training?
Actually, the plan is always there. It's just a question of yung resources talaga. Kasi yun we do have for example, what made na-mention ni Carlo kanina, 'di ba, yung internship training program namin, which is in Project and View. It was a 1-year training program, 'di ba? One of the things kasi that enabled ren people that enticed people to really stay there and go as part of the program was that meron rin means to make them stay there in a sense. Ayun, meron kaming binibigay na stipend. Okay. Actually, DOST nagbigay ng stipend kami na mumot. Ganun yan. Pero yun nga, at this point kasi very limited lang yung resources namin. We don't have a big of a financial bank account to be able to provide the stipend this time. Okay? So minsan nalilimit kami talaga to, for example, mga student interns talaga na lalo na yung kung saan required dun sa nila pag dito sa kanilang curriculum yung internship. So that's actually those are actually the roots of tag dito yung internship program. In terms of the trainings, we've been chalking up some sort of virtual trainings talaga. Although I don't know kung we're confident enough, okay, to do that. Okay. So that answers actually yung answer. Do you have training seminars online on bioinformatics? How to apply? Regarding sa training, we're still ironing out in details, especially dun sa administrative logistics part. Okay. Pero you could email pag dito. Ayan, where can we apply for training? So this also answers answer live. It's you could inquire then at bioinformatics at pgc.up.edu.ph. Ang major limitation lang namin dito is that of course it's gonna cost something. And also dependent dun sa need ninyo. And as much as possible kasi para medyo cost effective siya. I hate talking about costs, totoo lang, pero it's the reality of the thing. We try to parang host it or conduct it to a group talaga of people. So yun siya. Although at this point we're still ironing out the details, especially with the changes brought about by the pandemic. That kind of. Ayun. Yeah.
Ayan. So I guess we have covered the majority of the question. Yeah. Last question for Sir Paul and for the rest. No? What are the parameters in rejecting or accepting a sequenced sample? Quality control.
In particular for Illumina data, for example, you have what are called FASTQ files. So FASTQ files are actually sequence data plus quality base calling data. So for example, if you sequence a certain A in the FASTQ file that appears in the FASTQ file, you will have a base quality score. So for example, usually low quality bases start below 15 to 20. So if majority of your base calls are that low of a quality, so 20 or 15 below, then you can maybe reject that sequencing data. So you have bioinformatics software that can assess this.
Para sa mga totoong last na to, wait. Maganda tong question na to close our session. Dahil marami tayong mga attendees from Stratford. Marami tayong high school students ngayon. So, for high school students daw, ano daw po maging katulad nyo? Anong kailangan nilang gawin? Anong path ang kailangan nilang kunin? Course ba sa college to? Pwede bang hobby to?
That's an interesting question. Sige, can I say something? If you're still in high school and you're interested in this, well, for one thing, the nice thing is you can come into bioinformatics from many different fields, no? It's inherently a multidisciplinary field. Dr. Yap is from computer science. I'm from molecular biology. Polo, I can't remember kung ano yung background mo.
MBB.
MBB then. So we really come from, so you can come into it from statistics, from engineering, from physics, from classical biology, from medicine. So marami talaga. So if you're wondering about what college courses you want to take or what parang college program you want to enter, basically whatever related field is the most interesting to you. The other advice I have for high school students who want to become bioinformaticians or really any kind of scientist is to parang follow your passion and curiosity. In the case of bioinformatics, I would assume na may interest kayo in things like programming or numbers. So pay attention in your math classes. You can't, math is often the Achilles heel of any students. Kailangan magaling kayo doon. Learn programming. You can do that for free online. Learn Python. It's easy to, as programming languages go, it's easy to pick up. Maraming mga tools online to teach something like Python, even to children. Hindi kailangang high school. I have nieces that learn Python in grade school pa lang. So, and then as you build those skills, later on, when you have opportunities to get into actual bioinformatics, you already have you have a knowledge base to work off of. So, yun ang advice ko siguro. Baka maybe the others could give more illuminating advice than that.
Sa akin, yung journey ko was actually interesting. Okay, truth be told, I suck at biology talaga. If you're going to check yung high school grades ko, pakaramdam ko yun yung pinakamababa, even lower than art, and I suck at drawing. So, at some point kasi, siguro, and yun nga, computer science talaga yung background ko, right? Yung entry ko talaga sa bioinformatics was actually accidental, sa totoo lang. I just needed a job, another one, and then may research assistantship na in-offer sa akin. And then, later on, I guess what really got me hooked, I guess that's the word, into bioinformatics was that ultimately kasi yung computer science skills mo has to be applied in context, right? It has to be applied somewhere. And yung unang nilapit sa akin was that, ayun nga, ng advisor kasi Dr. Henry Adorna of computer science department, Steve Gilman. Nilapit niya sa akin was this mathematical model ng apoptosis. Now, apoptosis is, basically self death initiated by the cell. Pero ang naging come on sa akin, I mean, it's just a mundane thing, right? Pero pinag-aaralan siya kasi for to understand cancer, the mechanisms of cancer, and thereby trying to look at therapeutics and diagnostics. In particular, therapeutics kasi, yun nga, diba? The idea is you want to kill cancer cells, and you could do it by inducing apoptosis. And then it became a little bit personal because, yun nga, nung nasa PhD na ako, this is one of my untold stories. one of the things that really motivated me to do that was because yun nga, yung dad ko was a diabetic, and then um, I read a paper about talking about metabolic syndrome, and to a certain extent, I wanted to help out my dad. Fortunately, he didn't make it even before I finished my PhD. And so yun. Pero ang idea doon is that, ang naging focal point ko is that I found this to be something that would help other people. Okay, not only my family or not only myself. So yun. At this point, yun na lang yung naging ano ko eh. This is more on the philosophical level, hindi na sa technical. So it's more of trying to be able to contribute solidly to society itself. Okay, and I found that if I merge yung computer science ko with yung what I learned in computer science to field of biology and genomics and molecular biology, then it would be it's something that motivates me to be able to engage in it. So yun, at some point at the philosophical emotional um, level, talaga minsan you have to find motivation in what you're doing.
Yes, that's very enlightening, sir. Thank you for sharing. So I guess na-cover na natin yung topics na for presentation today. Nakapag-share na din ng ating mga panelists na experiences nila. Dahil alam ko marami nang nagugutom at lunchtime na, we would like to thank Dr. Jan Yap, Mr. Paul Gaite, Dr. Morao from PGC Mindanao, mga workmates natin, si Sir Carlo, for taking the time out of their very busy day to do this initiative of PGC. We would also like to thank the attendees. Yung mga nasa Facebook nanonood ngayon, shout out daw from PGC. And sa mga Zoom attendees, maraming salamat sa pag-attend. Maraming salamat sa mga nag-attend from webinar 1 hanggang webinar 3. We would like to inform you na we are still thinking of other series that we can do by the end of the year. Malalaman nyo rin yun by going to our website. So as stated earlier sa mga nandito, a link for the event feedback form is shared in the chat box. It will remain open until 2 p.m. today. So please answer it. Bakit kamo? Doon po namin i-base yung certificate of attendance nyo. And please make sure to read the questions carefully and paki-type po ng maayos yung name that you want to appear on the certificate and your email address as well para sigurado po tayong makukuha nyo na tama yung information. So we hope that the PGC Talks has helped all of us in learning more about COVID-19, from RT-PCR testing, whole genome sequencing, and of course, the bioinformatics part of analyzing the data about COVID. Siyempre, this is the third webinar. The first 2 webinars were already uploaded on our YouTube channel and Facebook page. Please, patasin natin yung views ng PGC. Gusto po namin talaga maging influencer. And we hope you'll join us again. Influence young people like you to become researchers and scientists. Siyempre, doon na tayo sa libreng information. For PGC update and announcements, please keep on following us sa Twitter and sa Facebook. Sa Twitter, it's PhGenome, and sa Facebook and Twitter, ganun din, PhGenome. We also have an official website, pgc.up.edu.ph. Sa website po namin, makukuha nyo rin po yung link ng PGC Mindanao, which will be launched soon, at yung website ng PGC Visayas. They also have special services for their specific regions. So again, this is Ate Shell, and this concludes this year's PGC Talks webinar series for COVID. Maraming salamat sa pagdalo. Goodbye, and magsa-sign off na po yung PGC Communications team. Maraming salamat.